Welcome back, everybody. So here we're getting ready for our second speaker of the day. We've got Dr. Bradford Casey. He's a Senior Associate Director in the Michael J. Fox Foundation's Research Programs Division. He manages grant projects and programs working with other Michael J. Fox Foundation scientists to manage the scientific portfolio and develop research strategies. Dr. Bradford began his research career working on the Michael J. Fox Foundation funded program to characterize animal models for Parkinson's disease and completed his PhD in neuroscience at UT Southwestern Medical Center, where he studied the role of transcription factors in neural development and cellular reprogramming. As a postdoctoral research scientist, his research focused on the development and application of novel genomic strategies in neuroscience, where he collaborated extensively with other researchers in neuroscience and cancer biology. In addition to academic research, Dr. Bradford has experience working with legislators and government agencies to advocate for neuroscience research. So his topic today for us will be genetics and Parkinson's. So welcome.
There we go. Hopefully you guys can hear me now. Let me just go ahead and share my screen. Okay, and if I could just get a quick thumbs up that you guys can hear me and can see my slides, perfect. That's the best way to start any talk, in my opinion. So my name is Bradford Casey, and I am really excited to be here today. This is a really unique opportunity for me because I usually spend a lot of time talking to scientists, and I don't very often get a chance to talk to patients and their family members. So I'm really excited today to tell you about something that I'm very passionate about, which is the genetics of Parkinson's disease. And I want to do so in a way that I hope that you'll all find to be interesting and maybe a little bit unusual. And so I want to introduce this in the form of a set of kind of historical stories, because really, when you think about it, genetics is unique in science, and that's part of the reason that I was drawn to it. So as was mentioned, I actually studied neuroscience in my PhD, but I really became fascinated by understanding how it is the different stories of genetics, which is really the stories of people and their families and the history of disease, comes together with some really interesting new technologies and new approaches to help us understand how that disease really influences or how that technology influences our risk for disease, how genetics can help us learn more about what's going on in a disease, and then ideally how it can help us address the disease. And so I'll go through a couple different stories, but before I do so, I just want to very quickly introduce what it means for me to be a scientist with the Fox Foundation. So the Michael J. Fox Foundation is a nonprofit based in New York. It was actually founded in the year 2000 by the Canadian actor Michael J. Fox. And we are really just an organization that's focused on we are dedicated to finding a cure to Parkinson's disease. And we do so just by funding research. And so we are a research organization. We collaborate with people around the world, funding people on projects big and small, to make sure that we're really going and getting into any aspect of the disease that is going to help us get to cures. And so that's what I get to do. I get to collaborate with lots of people to do so. I'm really excited to be here today to talk about it. Now, I know that many of you have seen a lot of introductory materials regarding Parkinson's disease, and so I'll just keep it very brief, point out that 6 million people worldwide live with Parkinson's disease today, and it's the second most common brain disease after Alzheimer's, and it is growing because people are they're living longer, and we're understanding more about the disease, and we're getting better at detecting the disease in patients. Now, it is a neurodegenerative disease. I know the last speaker spoke extensively about this, so I won't belabor the point, but I'll just point out that we know some of the clinical features of the disease, and we've actually known these for a really long time. There are a handful of these motor and non-motor symptoms, but all of those are really going to be considered clues here as we start to think about how we can understand the disease, what it can tell us about how we might be able to address it, and then also how it can tell us how it gets started. So first of the history lessons. So if we go all the way back to the year 1817, there was a physician living in London, and his name was James Parkinson. And what he started to recognize is that there were a number of patients that would come to the clinic. They would have a very common set of symptoms. And these symptoms were largely what we know about Parkinson's disease today. He identified the tremor that is associated with Parkinson's disease. He identified the slowness of movement, weakness in the body, as the specific stooped posture, and then kind of the irregular and short gait of a Parkinson's patient. Now, if I was to tell you the same update today in terms of how we describe Parkinson's, we would describe just a handful of other features, and they would either be very, very specific or they would be rigidity. So really, that's the only one that James Parkinson missed in the year 1817. So I point this out just to set the stage and point out that we have been working on this disease for 200 years in some capacity, and yet we are still working on it. And really, I think that the exciting aspect about this is where we're headed in that work. Now, a few years later, we got this illustration, and it's one that I see a lot, but I think that it's important, again, to recognize that Parkinson's disease is largely defined symptomatically, and that is one of the challenges about this disease. It's one that we don't have a great test for if you go to the clinic. There's a handful of behavioral tests. You might do things like the sit up and go test. There might be the POLL test that some of you may be familiar with, but we don't have what's called a molecular test. And so I'll talk a little bit as well about how genetics plays a role there. So when we talk about genetics, and we talk about heritable diseases, this is something that has been understood for a while. And often people don't really have a great sense of just what it means for something to be a heritable disease or a genetically based disease. But some of these phrases may seem familiar. And these are all phrases that I've heard from patients and their families. And someone in the family may have used to have a hobby or a passion or something that they were really good at doing. And then suddenly they just couldn't do that anymore. And they may not have ever been diagnosed with Parkinson's disease. They may have heard of the disease and know that it ran in their family in some way. They may have siblings or living family members that have the same problem or have similar symptoms. And so all of these are indicators that there may be some genetic component to a disease, but none of them are necessarily a clear indicator for a given disease, whether that is the cause or not. But the important thing to take away here is really just that there is a genetic link to Parkinson's disease, and that family history of Parkinson's does increase the risk of a person actually developing the disease themselves. So if we go back, again, looking through history, there's a handful of people that have really had just tremendous and outsized influences here. And I know that this will be a refresher for some back from your school days, but I think that it's really important to think about because it tells us a little bit about how we look at that information. So this is a picture of Gregor Mendel. He was actually, he was a monk and a mathematician and one of the early biologists in some ways. And he worked in his abbey, and one of the things that he did was he tended these pea plants. And he ended up publishing 2 papers on this and about really understanding how it is that if you cross different strains of these pea plants, that you can understand different features about them, and that there's a rule and that there's an order to how those features pass through the pea community, if you will. And so he identified that there were, for example, a couple different shapes of seeds, things like these round seeds or wrinkled seeds, different colors of both the seeds as well as the pods for these peas and even their flowers. And he did a lot of mathematics actually to understand how it is that shook out in reality as he started to cross these different pea plants. And I know you may be thinking, how is it that this is really relevant to humans? And the answer is, of course, that the same rules apply when we start thinking about humans. So, there's lots of traits in our family that we're all familiar with: hair color, eye color, height, things like this. And each of these may have both expected and then also surprising ways that they move through our families. And it's really, again, because we have a mixture of this genetic information. Now, if we fast forward just a couple of years, there was a man named Mishra, and he was a physician, and he was trying to understand a little bit more about certain types of disease. And what he really wanted to do is he wanted to purify these proteins, and he was doing so by working with patient bandage samples. So perhaps not the most exciting or pleasant way to spend a day for a researcher, in my opinion. But what he did accidentally is he ended up purifying this new substance that he called nuclein. And it turns out that that is actually what we call today DNA. And he didn't understand much about it. He didn't understand the structure of it. He didn't understand where it came from or what it did. He just knew that it was a new type of thing, and it was similar to the proteins in that it could be purified separately. And we didn't know what it did. Now, it took many, many decades before we started to understand that. And there was actually a couple of people shown here, Alfred Hershey and Martha Chase, and they did some really interesting experiments. And essentially what they did is they looked at viruses that are known to infect a type of bacteria. And they were trying to understand what it was that allowed the information to pass from those viruses into the bacteria. Now, I know the last couple of years have given all of us a better understanding of how things work. But what they ended up having to do is a really interesting set of experiments. And that is why there's a picture of a Waring blender. They became famous for these experiments because they really were able to uniquely identify that DNA is the specific type of molecule that underlies heredity. And even though they did this in this model using viruses and bacteria, we understood relatively quickly that this was also the case for humans and animals and plants. And so it really was a tremendous step forward in our understanding. And it really, because it was such a significant step, it was really just a couple more years before we were able to crack the genetic code. And so shown here are three people who are very central to this effort. James Watson, shown on the left, Francis Crick. They were working at the Cavendish labs over in Cambridge, and then Rosalind Franklin, who is working on something called X-ray crystallography, which is a way that you can use an X-ray beam, just like you would point at a potentially broken bone, but using a special type of X-ray microscope, you can actually start to understand the structure of these molecules. And ultimately, what they were able to do is understand the structure, and by understanding that structure, they were able to understand language. Now, they had different types of careers. Francis Crick stuck around in science a long time, did some other things, and actually worked with Rosalind Franklin, who unfortunately passed away quite young. James Watson actually left science entirely after some pretty disgraceful remarks, and so he's not really very fondly remembered. But they really made a significant contribution. And again, what they really came up with was understanding the structure of DNA. Now, the picture on the left here is what we all, I think, consider to be the cartoon image of DNA, right? Two specific strands of this molecule that are wrapped around each other in a very specific way, often shown at the front of every science textbook and at every scientific conference. And I think that people often look at that structure and they think, well, okay, so they figured out this structure, but why is that important? And the answer is that what they really figured out is how these strands pair together and how it is that a language can be wrapped up in this DNA, which is present in all the cells of our body, with only 4 biological letters, essentially, these 4 different nucleotides, A, T, C, and G. And the answer is that crack this riddle, that we have this encyclopedia long text of linear script, and all of the script works together, and we can actually start to read that. And it took us a long time to get to the next phase of understanding it, but these are fundamental steps that we needed to make so that we could really understand how it is that we went from Mendel's peas passing on specific traits to actually understanding how that happens in the cells of our body. So as a reminder, we're all made up of millions and millions of cells. Each of those cells contains an entire copy of the DNA for our body. So we have many, many, many copies of the DNA. It is present in structures called chromosomes, which are these long groups, and they're kind of the volumes, if you will, of that encyclopedia. And we and within those we have this DNA language. Now, the DNA itself is broken up into genes. We have about 20,000 genes that are relevant for humans, which is pretty amazing when you think about it. And each of those genes is made up solely of those 4 letters. And so that's wrapped up into these chromosomes and stuffed into the nucleus of all the cells in our body. Those genes are read by the machinery of our cells into something called RNA, which is similar to DNA. And from that, it is turned into proteins. And the important thing to remember here is that all of the instructions for every protein that will ever be made in our body is wrapped up in every cell of our body within that DNA, which is just a staggering thing to me. And you know, there's lots of comparisons about how long you can stretch it out and whether it can wrap to the moon and things like this. But the important thing to recognize is just that all of that biological complexity, from the rigid structures of the cells in our bones, the transparent cells in our eyes, and the perhaps in my opinion the most exciting, the electric electrically active cells of all the neurons in our brain, all of those proteins are wrapped up in this DNA and encoded. And anytime we copy one of those cells, there might be a couple of little mistakes. These are referred to as variations or mutations. And the important thing to understand here is that a tiny mutation at this step will actually be translated into the protein. So that protein will be forever altered in every copy that's made from that gene. So it's a really important thing to understand that variation. So there's a handful of different types of these variations. They're all relatively predictable when we really think about it. There's only a couple different ways that you can change the text in a book, right? You could accidentally have a typo and delete a letter. You could substitute one or more letters for something else, or you could insert it, which is, of course, going to separate things out. Now, the code of our DNA is broken up into little 3-letter words. So you can imagine that even a tiny insertion or deletion is going to shift all of those. And pretty soon, you might have the same letter sequence, but if the spaces aren't right, those words aren't going to make much sense. And so these can be tremendously powerful, even if they're very, very small. Some of these are unnoticed, right? They're the difference between a letter O and a number 0, right? They look the same to most people and to most things in our body. And some of these are going to be really, really deleterious. And so it's important to understand that there's a really broad range of how this plays out in our genetics. And understanding that is the first step to understanding how it is that we can all survive with a variety of different variants or mutations in our body, and then also how some of those can be really impactful at certain phases of our life. So before we go forward, I just want to pause, just have a quick summary of that. I know there's a lot of history there. I hope we've all found it as interesting as I do, but just as a reminder of the key terms, the DNA is our genetic code in each of our cells, right? These genes are the words there. They're these inherited bits of information that influence who we are. And then the variants are the changes in those genes, and a mutation is specifically a bad variant, right? Is a variant that is indicated or is tied to a specific risk or disease. And so it's just important to understand all these concepts as we start to move forward. Now, so far, you've gotten a little bit of a lecture about genetics, but you haven't learned anything about Parkinson's that you didn't already know. And so next, I want to talk about what it is that actually causes Parkinson's disease. Now, as you know, for most people that develop Parkinson's disease, they may not have a family history. So there's a lot of people that are going to kind of fall in this middle category. They're diagnosed with Parkinson's. They don't know much about how they got there, but they know that they're there, and they want some answers there. And we think that there's actually a variety of different causes of Parkinson's disease. And in fact, we now really talk about Parkinson's disease more as a collection of diseases that have a similar outcome, again, the symptomatic outcome. And they have these different causes, but the important thing is to understand how they're similar, how they're different, so that we can eventually get to cures and make sure that we're getting the treatments in the hands of patients. So in addition to just a general, let's say a potentially random risk of Parkinson's disease, we know that there are a handful of environmental factors. So exposure to things like some pesticides, repeated head trauma, these are things that have long been associated with Parkinson's disease. And for a long time, it was thought that this must be the primary risk, and there was some random risk and then these exposure risks, but we didn't really know as much about the rest of it. But in the last couple of years, because again, this technology has gotten so much better, because we've learned a lot more about these stories and about the data that is available for genetics across disease, we started to uncover some other things. And just in the last few years, really, we started to understand these genetic factors a little bit better. And today I'm going to give you 3 vignettes about 3 of the most important of these. Now, there are many more, but we don't have that much time today. And I really wanted to just pick these because I want to talk to you a little bit about how it is that these are really, I always think of these as being kind of examples like we might have found many years ago in an adventure book, right? An Indiana Jones movie, for example, because there's stories that really require you to think deeply about the problem, go around the world to find the answers, and bring together all those clues, talk to all the right people to make sure you have a whole view of the situation. So, there we go, a little faster. So I want to just briefly talk about something called the Parkinsons Jar model. And this is, I think, a really great example. I didn't come up with it, but I think it's a really great example to kind of compare these two different forms of risk. Now, one of them, again, is these environmental risk factors, right? Looking at how that might have an interplay. There's also these genetic risk factors that really are present in all of us from the time that we're born, right? We don't know just what we'll have. Some of them might be very large. Some of them might have a very small influence, but ultimately, we all have this Parkinson's jar. And if we look at it, we start with it being full of only these genetic factors, and over time and exposure and risk, illness, aging, all these things, right? We start to build up these exposure, these environmental factors. And ultimately, whatever the cause is, whether it's primarily the genetics at the bottom of the jar here, the things we were born with, or the things that we were exposed to, eventually you develop Parkinson's once that jar is filled to the top. And there may be people who have lower risk, either because they just have a taller jar here, right? Or they may have fewer genetic factors. And so they may never in their lifetime actually develop enough of the damage that goes along with it to experience Parkinson's disease. And so I just think this is a really helpful way of thinking about that interplay between the genetics and then also the environment, which is really important for understanding disease. So I want to show you this figure from a paper, and this is from about a decade ago. And essentially, while this was not a new idea, this is a great way of sharing it. And so you can think about this in terms of how these mutations come together with the environment around us and how severe they are and how common they are. So mutations come in many forms, and it's really important to think about how strong the severity of each individual mutation might be. Over here on the left, this is kind of indicated as severity. It's something that a geneticist would consider the effect size, which is basically how effective that mutation or that variant may be in changing something biologically, including the development of Parkinson's disease. Now, for Parkinson's disease, we have a handful of these that are going to be relatively rare in a population. You're not going to just randomly go down the street and find lots of people that carry them, but they're going to be very strong at causing disease. And they often tend to be more following these Mendelian ratios, right? Again, the very direct heritability. And when they do show up, they're very likely to have an effect. There's also some that are perhaps medium or lower frequency, but they have relatively strong effects. And so these may not happen until later in life. People might get into their 60s and 70s before they start to see the effects of these mutations. They may be more likely to be influenced by environment, things like that. But it's important to understand that there's a range of strengths of these things. There's a range of the severity of any individual mutation. And in fact, many people who have these genes may never see disease in their lifetime for a variety of reasons. That takes us to how we can actually study these genetics. And surprise, surprise, it's as I mentioned, it's really a story about families, right? It's a story about how these genes pass down. And so just as Mendel tracked a trait from the first generation of a pea that had a yellow pod into finding that all of the progeny, even when crossed with a green pea, led to a yellow pod pea, right? That if you go down the rows a few more generations, you start to see those green peas appear again. And it's the same with different forms of genetics of disease. And so this is just an example of what we call a pedigree. And if you look at it, you can see that at the top, there's a male and a female. This line indicates the female's passed away by the time we're doing the analysis. And you can look down and you can try to find the specific trait, whether that's disease or just a feature like eye color, and how that actually follows through the family. And again, I point this out just to set the stage for these detective stories, because as I said, you have to be really willing to go and dig deep and understand this. And that's a really important piece of the science behind the genetics of Parkinson's. So the first, I think, is one that is really fascinating. So again, Parkinson's was discovered in 1817. We didn't understand the genetics of Parkinson's, or we didn't understand the genetics of anything really until the 40s, the 1940s, 1950s. And it wasn't until 1997 that we started to have these really clear examples. And that is an interesting feature, right? Again, that tells us a little bit about the disease. But these are, again, if we were to look at that continuum, these would be in that far high severity, relatively rare, very rare forms of the disease, and this would be one of those. And so essentially what happened is that there were some researchers that were trying to understand this. They went out and they looked at different patients that were developing Parkinson's disease, and they found a very clear familial case, a case where the family was all developing. Many of the people in this family were developing Parkinson's disease. And they went back and they looked through their family records, and they went and found all the people that were still alive. They went through and they talked to as many family as they could, understood, well, what about if you were married? Did that person's family have Parkinson's disease? And then ultimately they came down and they said, okay, so we have this very good chart. So again, you'll see that this is just a longer version of the chart I just showed you. This is a five generation pedigree. And so they identified all the individuals here that have the disease shown in black in this plot. And they then they took that and went back to the actual genetic information. And they looked at all the little genetic clues. And again, there's millions or billions of these different clues that are present throughout our genomes, which is just the genome is just the word for the entirety of our DNA. And so then it becomes a puzzle, right? You have to figure out which of those clues only follow the disease and which of them are kind of present in one form or another, regardless of disease. And when you get to that smallest set, that is the really informative piece. And what they did is they eventually found out that there was this specific set. This is called the Contursi kindred. And they found that they had a specific substitution mutation that was present in a gene called alpha-synuclein. Up here, SNCA stands for alpha-synuclein. All the genes have these names. There's 20,000 of them, as they said, and they're a little tricky, and so they've made them pretty short. And anyway, so the exciting piece here is just that they were able to find the first monogenic case, the first thing where one specific variant was tied to this disease. And these types of monogenic cases are relatively rare, especially in Parkinson's disease. You're not going to find a lot of Parkinson's patients that have this level of severe mutation, but they are out there, and they're more common than you might think. People with some of these very strong mutations tend to develop disease a little earlier in life, which is important. And they tend to have a little bit more severe disease for a variety of reasons. And again, there's many different variants, but this is just one of them. Now, synuclein in particular, again, this SNCA gene encodes a protein, as I mentioned. So all the genes in our DNA turned RNA, turned into a protein. And so this mutation is encoded into that protein. And it just so happens that the same year, 1997, a different group was looking at these different features. And what they came to understand is that Lewy bodies, which were discovered in, I think, 1912, these were little spots in the brain. And we had known that they seemed to be present in people that had neurological disease, perhaps Parkinson's, perhaps other diseases. And we were trying to understand that. And so an independent scientific strategy was to understand what was in these Lewy bodies that seemed to be a piece of Parkinson's. And then they were able to identify this, and it turns out that this was the same exact protein that was found in the brains of those patients. Now you might ask, well, I thought you said that this mutation was rare, and it is. And that's a really important thing to take away, is that even from a very rare mutation, we can learn a lot about the significance of something that affects a much broader picture. So looking at these cells here, they're indicated by the arrows, and then the fat arrows indicate little pieces of the cells called neurites. These are cells that are dying or have already died, and turns out that they have a lot of this alpha-synuclein protein that's in them, and it appears to be evidence of the damage. We still don't fully understand the role of alpha-synuclein. We have a lot of work going on there to understand that better. But what we do know now is that if we see Lewy bodies in the brain, alpha-synuclein is going to be a strong piece of what makes up those Lewy bodies, and so it's a really clear indicator that Parkinson's pathology is going on. And I've included this text not because you should read the whole thing, but just to point out that these Parkinson's disease cases that were studied in this study, showing these images, these are non-familial cases. So these are not people that have that specific mutation. And yet we still learn so much about the disease from seeing it. So again, just really important to understand that for every one of these genes, it has a story. And our ideal vision is that by putting together all the clues, not just from a single family, not just from a single gene, not just from a single mutation, we're able to actually build out that bigger picture of all the things that are going on. So next story is a gene called GBA1. And GBA, it includes a specific type of enzyme in the brain, and it's a really important enzyme, and we know that because people, as I mentioned, we have 2 copies of each of these genes. If you have 2 copies of this mutation, this Gaucher's disease mutation, you develop a disease called Gaucher's disease, and this is a really devastating disease. It strikes people very young in life, often just as they're 2 or 3 years old in some cases, and most of these people do not have an opportunity to live very long for a variety of reasons. It's very hard to treat, and the treatments are not super effective for some of the pathology. And so ultimately, it's a very damaging disease. And we've known that for a while, and people have been aware of and have sought to understand the biology of Gaucher's disease. But it was always understood that you needed to have these 2 mutant copies. You have to have 2 bad copies of the gene to develop the disease. And even though there's a couple different forms of the disease, understanding that you need those 2 bad copies was really important. And so it was thought that this gene that encodes a specific enzyme is responsible for this disease. And then we started to do these bigger studies, and we started to realize that some people that had Parkinson's disease had 1 bad copy, just 1 mutation. And it seems that those people do develop Parkinson's disease, but they still develop it relatively late in life. And so that's what this paper is from. So this is a paper from 1985. They started to recognize that there were people that did not seem to have what we would consider to be Gaucher's disease, but they had another disorder that had some of the same features as Parkinson's disease, specifically dopamine responsive, what's called an extrapyramidal syndrome, but is essentially the same characteristic phenotype of tremor and weakness and stiffness. And so really it was the same presentation as Parkinson's disease. And when we started fast forwarding a few more years and doing more analysis, it does seem that this is a subtype of Parkinson's disease that does show loss of the dopaminergic neurons. It does show changes in the brain that are similar to Parkinson's disease. And so this is a really important clue for us that tells us that maybe in addition to synuclein, GBA could be a gene that is involved in Parkinson's disease. So I mentioned previously that there are 20,000 genes that we each have. We each have all of those genes. So we each have a copy of synuclein in our body. For most of us, it's operating fine. It's a function of our nervous, or it functions in our nervous system. We also have a copy of GBA1. And so it's important to understand that all those genes come together in this big complex machine, far more complex than any wristwatch that the finest watchmaker could ever make, right? And I think it just goes to show that if there's a one little break in one little gear in that giant wristwatch, that it can affect things all over. And so it's important to understand that, because you start to understand, well, that watch or that gear touches this gear, that gear touches this wheel, that wheel touches, the date or some other function of the wristwatch. And so ultimately there's a lot of different ways that things can be broken that can lead to disease. And that takes us to the 3rd example that I want to share. So this is a gene called LARK2, another gene that is linked to Parkinson's disease. This encodes a protein called leucine-rich repeat kinase, which I know is a long name, and it's not too relevant for today's discussion. But I like this because it really highlights the process that scientists use to understand that, to go through this detective work and really understand just which piece is going on. So if we look here, this is a study from 2002. And they were looking over this case of another familial case like the synuclein, but for a different gene. And they had found it in this Japanese study in 2002. And so if we look here, this is a map of one of the chromosomes. Again, we have 23 of these chromosomes in our bodies, and 2 copies of each one. This Japanese study, even in their first analysis, where they identify that, okay, this appears to be linked to Parkinson's disease, they were able to narrow it down to just one small piece of one of those 23 these chromosomes, again, these books in the encyclopedia, and they were able to decide, okay, somewhere in this region, there's a change, and that change appears to be the piece that is linked to this disease, to Parkinson's disease. And so then within the same year, a different group was looking in a different cohorts. They weren't looking in Japan, they were looking in northern Spain, the Basque Country, and they found a different group of people, and they said, okay, so somewhere in this region, it seems that there is a mutation. And so then they combined these 2 analyses in this paper, and they said, all right, so we're going to try to find this in even greater detail. And they eventually narrowed it down to just this tiny little fraction of just one of those volumes of that big genomic encyclopedia. And they said, okay, within here, these are the genes that are present. I told you there's 20,000 of them, and they all have crazy symbols and everything. But within that 20,000 genes, they were able to identify that it is in just somewhere within this set. So again, it's this long process of narrowing it down, of identifying those clues using the biology around us, traveling the world to find the people that are affected by the disease, and then ultimately going back and doing the careful, quiet work of studying in a library and trying to understand and bring it together. And that does involve a lot of experimental work that I won't go over today, but it's important too. So from that, we said, okay, and this I would point out is a few years later work, but essentially we were able to identify this LARK2 protein, again, the leucine-rich repeat kinase, which is an enzyme protein. And if we look at it scientists were able to actually understand, like, okay, so this protein, like many, is made up of these different domains, these different regions of the protein that may have different functions. And they started to look around. They said, okay, so there's the Japanese cohort that we found. There's this Basque kindred. There's a group of these people in North Africa called the Arab Berbers, right? And all of them are developing some form of disease that appears to have to do with it. And so they started mapping where the different variations, these different potential mutations to cause Parkinson's might be. And so these are just the variants that have been identified within Lark2, and this was as of 2020, and this is even just a handful of them, that appear to be associated with disease. So some of them are in what's called the armadillo domain. I'm not sure why it's called the armadillo domain. This is, by the way, this is a beautiful molecular image. The one thing about being a geneticist is you don't get to work on the structure, so you don't get to see these beautiful images as often. But so this essentially is this is all one long protein, one long strand of amino acids. It has both the real structure, which is again determined by the X-ray crystallography or similar approaches. Same thing that was used to understand the original structure of DNA. And then there's a cartoon just to help us keep it all straight together so I can go through this with you today. And so essentially each of these mutations, right, each of these potential causes of Parkinson's disease localizes to a specific piece of this structure. And that is an important thing. And that's part of the fun of this puzzle, is that we're not just looking at numbers on a page. These are real things that are present in the cells of our body. They're in there. These enzymes are doing things. They're adding phosphorus to things. They're binding to things, cutting molecules apart, all the different things that enzymes can do. And so it turns out that there's a handful of these that are very significant. I know there's a lot on this slide, and I'm going on, but I think it's a really exciting story. So each of these, again, is one of these mutations that is potentially causing disease. Each of these corresponds to a change somewhere in the actual structure of this protein. And so then the magic happens of trying to understand what is actually going on, where that biology is happening. And it turns out that if we actually look at where all of these are across the length of this protein, a lot of them tend to bind down here in what's called the kinase domain, or this ROC domain. And so these are parts of the protein that are the enzymatic parts. And so that tells us that the reason that these people from a variety of different places that are getting essentially the same disease are getting that disease appears to be that we are changing the activity of this enzyme. And ultimately, when you get to something like this is a tremendous discovery, because that is where we can start to think about drug discovery. That's where we can start to take this to the next step. So these are the three examples of this monogenic disease. I also want to talk about some of the other pieces. So I've told you about these rare alleles like synuclein that cause this Mendelian disease, where if you have the gene, you're very likely to get it. You're likely to get it younger. I've told you a little bit about some of these things like GBA and Lark2 that may not lead to Parkinson's disease in every patient and may interact more with things like the environment. But I haven't told you about these common variants that are very hard to track down because they require a newer technology to really look into it, and how we can look into that, and how that is an important piece of our understanding of Parkinson's more broadly, and also how that's important in our strategy to address those different things. So the important thing here is that this is called complex genetic disease, and many variants of smaller effect basically come together to potentially cause it. So I'm going to show you a new way of looking at this data. So I've told you that we have these 23 different chromosomes. If we spread them all out, so this would be essentially like taking all the books and spreading them out on the floor so you can see the genome from book 1 all the way to the end, right? You can take this and you can look at the position of the genes. And so what this is an image called a Manhattan plot. It's called a Manhattan plot because basically it's a way of looking at important areas of the genome that are focused on a specific disease or specific outcome. And it's called a Manhattan plot because if you think of it as a Manhattan skyline, right, just stretching out into space. In the old days, the taller the building it was, the more important it might be, right? The Empire State Building, right, a pinnacle of industry and things going on. The Chrysler Building, again, a big company building things. And so these really important areas in the genome basically had a heightened importance for a specific thing. Here, looking at Parkinson's disease. And so if you were to think of those as kind of books in a library, right? And each one would have a card in its card catalog. Every time that book gets checked out, it gets stamped by that librarian very diligently. And at the end of the year, if you were to take all the cards in the card catalog out and look and just count up how many times each book had gone out, you'd find that there was a lot of books, these ones that are very low, shown here in gray, right, that hadn't been checked out any times or many times. And so if you instead were to say, well, show us the ones that were checked out by everyone, which would be, for example, the ones that were checked out by every patient with Parkinson's disease, right? Those would be books on these genes that seem to cluster together, these books about a specific topic. And so each of those can kind of come together. And so this is really just a way for scientists to look at this genome-wide, again, all the chromosomes at once, and understand those genes that appear to be the most significant. And so this image is actually taken, a friend of mine named Mike Knowles and many other people worked on this. And they did this back in 2014, and they looked at all of the data that was available at the time, which at the time was 13,000 patients with Parkinson's disease, 95,000 controls, so people who did not have Parkinson's disease. And together they said, okay, if we look at all of this data, which are the ones that are only found in patients with Parkinson's disease? And they ended up identifying 26 different sites. These are called locuses, across the genome. And these are the ones that are thought to be the most significant. And I know that you're asking why am I showing you this plot? It doesn't really tell you much about the science of it, but I think it's really important because it highlights the progress here. And so the next slide I want to show you is this one. So this is just 5 years later. And within those 5 years, we were able to expand this to 37,000 patients with Parkinson's disease and over 1.4 million people who did not have Parkinson's disease. So you can imagine the power of that. If you're trying to just find the clues that only go along with disease, you really need these big numbers. And so just in that 5 years, we went from 26 loci that are significant for Parkinson's disease to over 90. And each of those loci has its own story. Each of those helps us to understand that whole picture of how it is that an individual gene or a section of genes, a collection of genes breaks, how that is going to affect patients at the end of the day. So I think this is really exciting because it really highlights the progress that we're seeing here and how important it is to get large numbers together. And in fact, that progress, that collection of these large numbers is so important that we are now seeing even bigger efforts. So this is a slide I'm really excited about. Just an overview of what's called the Global Parkinson's Genetics Program. This is something that Fox is involved in, as well as our ASAP partners and a variety of people. But the important thing is that this is not just us. This is people around the world. So from over 57 locations around the world. We have 130 different cohorts. These are groups of people. These may be scientists, these may be physicians, these may be hospital systems. And what they're doing is they're working with patients to donate the genetic data from their studies to help us understand Parkinson's disease. And we just so far have over 160,000 samples that are promised that people are willing to share. And so again, getting those numbers up is a key piece to making sure that we go from those 90 ideas to those 90 understandings, and from that 90 to the next 90, right? We want to have all of this. We want to have it all basically as soon as possible so we can actually develop these into cures, get them out the door. That's a really key piece here. The other important thing to understand is that this is a global effort for a reason. It's not just that we want more people. A lot of the history of genetic studies has really only been done in a couple of places: Northern Europe, US, Canada, and all of these people have very similar genetics if we look at the ancestry. And so understanding this from a more diverse perspective is super important. One, it will tell us more about variants that we don't see in Northern European populations. That's one piece. Two, it's going to tell us more about how all those pieces work together to understand the genes between them, how they may be arranged slightly differently, how common they are around the world. And then three, ultimately, this is going to make sure that the cures that are developed are going to be effective for everyone. They're not just going to be effective for a group of people that are very, very closely related. They're going to be effective for as many patients as possible. So it's in everyone's best interest for us to expand around the world. And while it's very early days for this project, it's tremendously exciting. And I'm just thrilled to be able to share a little bit of it with you today. Okay, so I've told you a little bit about the history of genetics. I've told you a little bit about the history of some of these specific detective stories, these specific genes that we're interested in. But now I want to tell you about why it matters, right? I want to tell you about why it matters to patients. And the answer is the genetics, again, they really give us a starting point towards these new treatments that are going on. So this is just the first stages of this. But the idea for any genetic study is basically to identify these mutations that are associated with Parkinson's disease. Then you can identify the protein or the change in something else that's going on that might be caused by a mutation like that. And ultimately, you take that information and you directly take that to develop treatments that are going to prevent that are going to fix it, that are going to help people understand their health and what may be coming up for them in the future. In addition, these genetic studies are really important because they will help us to understand these broader aspects. As I mentioned, alpha-synuclein, right? It was discovered in one family. one family, but it matters for every family that has Parkinson's disease, because these alpha-synuclein clumps tend to form in the brains of patients even that do not have this mutation. So information from a single family, a single study, a single gene, they really benefit our broader understanding. Again, thinking about this as this fine Swiss watch, right? We need to know how all the different genes may break, how all the different gears may fail to come together, and that's going to ultimately tell us more about the disease. So some of these treatments that we're working on now are actually going to be personalized. And the thought is that basically there are certain people who have a certain type of Parkinson's disease, and we may be able to treat them with one kind of medication. But there may be other medications that are really going to work for everyone or are going to work for a different subset. And we want to make sure that every patient gets the right medication to actually treat them, right? That's the most important thing. And so this is a concept called personalized medicine, but essentially a lot of the recent trials are really looking at genetics as a way of improving their understanding, but then also improving our ability to target that treatment so that we can make sure that everyone gets the right medication at the right dose at the right time to stop, perhaps delay, perhaps even prevent Parkinson's disease. It also helps us to understand the role of the environment, right? Because if we know that some people are just going to get Parkinson's because of their exposures in the military, for example, that's one kind of Parkinson's disease. Maybe we need special treatments there. We may know that there's other people that have the specific risk variant, and they're fine unless they have certain types of that exposure, right? There may be people who have a variant that is just highly damaged by sun exposure or highly damaged by exposure to a certain chemical. And so understanding that tells us more about the mechanism by which it breaks, but then also how we can make sure that that's included in our overall understanding of the disease. And ultimately, genetics is going to also be used to help select people for studies and make sure, again, that we're building out the right studies that are testing the right question that are actually going to help us drive this field forward. And even right now, this is not just in the future, this is already happening. There are certain studies that are going on in the US and Canada and around the world that are really targeted towards these specific genes that I've talked about today. And there's more than just these. I want to point out this, I had to cut this down to make it all fit on the slide, but this is just a handful. And you can learn more about all the different studies that are going on our website. But just want to highlight that we are already looking at certain types of people's genetic backgrounds to help make sure that we're putting them in the right trials, that we're testing those right questions, that we're ultimately going to make the most of the studies. What else can we learn? Another piece that is a real challenge, as I mentioned up top, we don't have a test for Parkinson's, right? Isn't that crazy when we think about it? It's a disease we've known about for 200 years. And you can't go and have them take a drop of blood. You can't go and have them take a little bit of urine or even take a picture of your brain and really understand whether or not you have Parkinson's as a clinical test. And that's really hard on patients and their families as they're trying to move through early stages and learn whether this is really Parkinson's, whether it's something else, help us understand what kind of disease it might be. And so this all comes down to a concept called a biomarker. Biomarkers come in a lot of different flavors. One would be something like a blood pressure test, right? You can go in, they'll pump up the cuff on your arm, tell you if you have high blood pressure, you might be treated for that, come back in, check it again and see if it's changed, right? That's one. Another would be something like a hormonal pregnancy test, right? You can basically put some urine in a cup and a chemical test will tell you if you're pregnant based on the presence of specific proteins, these specific hormones that are present in the urine. So these come in a lot of different ways, and it's really important to understand that if we can get that if we can develop that for Parkinson's disease, we're going to be able to run these trials faster. We're going to be able to run better trials to understand, are we seeing changes in response to a drug? And ultimately, as we look from today towards the future where we may, in addition to having treatments like levodopa that are going to help with the symptoms of Parkinson's, we really want to be curing Parkinson's. We want to be coming up with treatments that are going to actually change the course of that disease. And so this is going to be a really important piece if we want to understand how a drug changes that course. We need to find these biomarkers and we need to make sure that we're following them forward. So now I've told you a little bit about the history, told you a little bit about the stories about these genes. I've told you about the importance of understanding these genes, and you, I hope, are interested in the idea of at least what it might look like to get genetic testing. There's a couple different ways that you can access genetic testing, and for patients that often comes in the form of access to research studies, right? This is a situation where you might go into your healthcare provider and they might say, oh, there's a study about Parkinson's, are you interested? They might swab your cheek, ask you to sign some forms to share the data. And ultimately that there'd be a researcher, someone like me, basically, that's going to be on the other side, that's going to take that data, and we're going to try to go through it and understand it. And in some studies that involves some return of results to patients to let them know what we found. In some, it's more exploratory. We might be looking at genes that are not yet associated with Parkinson's disease, and so you might not get that data back. There's also clinical genetic tests. So this would be something like if you go in and you say like, oh, my grandfather had Parkinson's, I'm starting to have some symptoms. Your doctor might say, aha, that sounds kind of like one of those pedigrees, one of these familial cases. I should submit this for genetic testing. Right now, we don't change the treatment strategy. So this is a little bit less common, but we think it may change as some of these treatments are targeted towards different patient groups. But that's nice because your physician may be able to submit that test, put you in touch with people that are going to help you understand that information and perhaps help you get a diagnosis a little bit sooner. There's also things like direct-to-consumer testing. You may have seen these kits on the internet that'll tell you a little bit about your genetic ancestry, or may tell you a little bit about your drug risk. There's obviously a couple different providers. 23andMe is one that we often get questions about. And these are good options, but it's important to recognize that each of them has kind of its own focus. The information that you might get from the test is going to vary quite a lot from these different options. And ultimately, you're going to need one that's going to work for you and potentially work for your family. Some of these are going to come with what's called genetic counselor option. And I think that that's a really important piece, because for most people, to be honest, even me, a person who works in genetics a lot, I can't really interpret the significance of all the results for diseases that I may not study directly. So these genetic counselors are really out there in the field, understanding this and understanding how it impacts patients. And they're experts at making sure that you can get the test that you need, that you can interpret the results of that test, and ultimately you can make the most of the experience. So I think anyone that's interested in genetic testing should be asking about access to a genetic counselor so they can help make the most of that opportunity. Oh, okay. So now I'm going to tell you a little bit about something I think is really exciting, which is how you can be a part of the solution, right? And I think that it's important to recognize that people with and without Parkinson's are both part of that answer. And I told you about some of the crazy numbers that we need to get to those next steps. And so we do need people that have Parkinson's disease, but we also need people that don't have Parkinson's disease, especially people that might have it in their family or might be at risk for Parkinson's disease for a variety of reasons, but may not have it. And that is going to be a really important piece. And so again, depending on your background, depending on your genetic status, you might be able to participate in different forms of research. And I think that that's a really exciting option that many people don't realize that they have. You don't necessarily have to take a drug to be an important piece of our changing the understanding of Parkinson's disease. And that's going to help us understand these new genes that are emerging, understand new genetic links and how these mutations are going to be important to fixing the problem. It may be important to understanding how genes impact which type of Parkinson's you get. Like I said, there may be some that are more depend more on exposures, things like that may depend or may tell us a little bit more about when you get it or how you progress through the disease. And ultimately, this is the next step. This is the limiting factor for how we develop and test new treatments, right? So important to recognize that you don't have to take a drug just to be involved in a trial, but there's a lot of different types of research going on, and we definitely encourage people to look into it and get involved. So if you want to be part of one of our flagship offerings, this is something that I'm tremendously excited about. So way back in 2010, I know that doesn't seem like a long time ago, but it feels like a long time for me. We launched a study called the Parkinson's Progression Markers Initiative. And the goal of this is to understand and validate these biomarkers that I mentioned for Parkinson's onset and progression. And it was a really effective study. And in fact, we have seen almost 10 million people download the data from this study. So I'll tell you, I've worked on a lot of projects in my life, and I'm not sure that any others had 10 million people interested enough to download the data that I generated. So it's really, I think, an important example of how important this is and how broadly this is needed in our field. And this is a project that was really designed to understand people's progression through Parkinson's disease. And so we identified people who are at risk for Parkinson's disease based on a couple of specific factors, and people who are just being diagnosed for Parkinson's disease. And then we followed them for a few years. And some people have been in the study for, I think, a decade now. And we routinely go back to them. They provide biosamples like blood throughout the process. They also get imaging data that we collect, so MRI, and then something called a DAT scan that some of you may have heard of, looking at dopamine in the brain. And our goal is to understand the progression of Parkinson's. Again, surprise, surprise from the name. But really tremendously exciting. And the important thing to take away here is that just in 2020, we launched an expansion of this study. So we are now expanding from just 33 sites to 52 international clinical sites, and that number will actually probably go up throughout the study, including sites in Canada, I want to point out. We are growing from 1,400 patients in the original study to over 4,000 patients that are going to see clinical visits. We are launching the PPMI online component with the goal of gathering data from 100,000 people who may not be able to go in for the clinical visits, may not be candidates for the imaging or something like that, but still have something to contribute to the study. And so there's 4 sites in Canada right now that are recruiting for PPMI. So it's certainly something to look into if you're interested. They're specifically looking at recently diagnosed patients, people that have Parkinson's risk factors, including family history, including something called REM sleep behavior disorder, where people act out their dreams in their sleep, and then also known genetic links, which you would discover through this type of genetic testing. We're also looking for control volunteers, people who don't have Parkinson's in their family, don't have any clear indication of disease, but are in the right age bracket, because again, that control data is super, super important to helping us understand just what's going on. And we've had patients that reach out to tell us about their experience with PPMI, and I always think it's really interesting to hear about it. I've had people say, like, look, I went into the doctor and they said, oh, I've been having these issues. I'm acting out my dreams. I punched my partner in the face, when I was sleeping. It's a real problem for me. And they said, oh, that's interesting. It could be this, it could be this. They did some testing, and ultimately they were referred to PPMI. And now that same person is getting involved in the PPMI trial. So I think it's just an interesting example of how people come from all walks of life and all different backgrounds to help join us and be part of the research, which is obviously so important for the work that we do. Okay, so just to wind down, I just want to go through a little bit more about what I told you today. I hope that at the end of the day, we'll be on the same page. But in summary, genetics is a key piece of our understanding of Parkinson's disease, and it informs how our DNA shapes our risk for Parkinson's disease. There's a lot of different genetic variations, mutations, which affect this risk. As I said, we have at least a handful that are strong, some are weaker, and we have a lot more that we can't wait to discover the secrets of. It represents only a fraction of that total risk for Parkinson's, things like environment and especially age, obviously. These are all key pieces, and they are all a factor in your risk. So genetics is not the only piece, and if you have a gene that's linked to Parkinson's, you may not ever develop Parkinson's, but it's helpful to help understand how that risk affects you and what it may mean for you moving forward. We are focused very specifically on studying these genetics to help understand Parkinson's disease. And our goal ultimately is just to develop tests, treatments, and cures, because that's what we're here for. We're here for patients. I think the entire scientific community would agree with that. Our goal is not just to add more to that list of 90 different loci. Our goal is to make sure that those are translated into something that's actually going to affect you in your life. And so really excited to get a little bit of an opportunity to share this information with you. We just finish up by saying that, again, genetic research is really a team effort. So it's not just scientists like me, it's patients around the world. And ultimately the goal is to benefit patients that do and also do not have these specific mutations. And we're really excited to be partnering with the community to help understand that and help make sure that those benefits are shared with everyone. And ultimately, I just want to kind of end on a note and just say that I'm really grateful for the opportunity to talk to you today. There are a lot of things that you could do with your Saturday, and this is just one of them. And you today have chosen to show up and learn, and I think that's really, and really that is your first step as a student of genetics. So congratulations on being a student of genetics, and thanks very much for your attention, and really appreciate the opportunity and hope that I'll have an opportunity to answer some questions here. But again, really excited to be here. A couple acknowledgements and just people who have contributed to my presentation today, and then also funding this is largely provided by ASAP for some of these programs, GP2, again, the Global Genetics Program that I spoke about, and then obviously the Fox Foundation, and of course you, the Parkinson's patient community, as well as caregivers and physicians and everyone else. So thanks very much.
Wonderful. So yeah, we've got a couple minutes here for questions. So the first one here, are any of these PPMI genetic research opportunities open to Canadians, or are any of them being run by Canadian research groups?
Yeah, great question. So there are a number of studies that are run by Canadian research groups around the country. And I would just point out again, we fund a lot of work that goes on in Canada, all the way from at least from Montreal to Vancouver, probably even further if I looked at the map a little more closely. So there may be studies in your area, which I think is a really great way to get started. And you can always go to our website to learn more about the work that's going on, where that work's happening. For PPMI specifically, yep, we have at least 4 sites that are recruiting in Canada, so that's really exciting. Obviously, Canadians are patients too, and we want to make sure that you're represented in the research and all the data that comes up.
Excellent. I have another one that kind of follows that up. So with these PPMI studies, how far removed from Parkinson's disease do you have to be for like the family genetic studies?
It's a good question. So how far removed do you have to be? So if you want to be a control, they would probably ask if anyone in your family had Parkinson's disease or had ever tested positive for a genetic variant that was related to Parkinson's disease. For some studies, they would be looking at a broader pedigree, again, trying to understand your family more broadly. And so if possible, they might go back and ask like, oh, do you have siblings that are interested in participating in the study, things like that. It really just depends on the study and depends on what it's trying to understand. There are some studies that are very focused and they're really trying to understand like the significance of a gene more broadly. And so I think that it's an important example that just, there's something that can be a fit for everyone. And even if you don't end up contributing, genetic samples or anything, biological samples, you may be a fit for one of these online studies that helps to just understand how it may be present in a certain population. As I mentioned, a lot of these are really history stories, and they genetics really tells us about how it is that humans move throughout the world over history. And so a lot of it comes back to ancestry. There's certain ethnic groups or ancestral groups that have certain predispositions and may not have others. And so those are groups that we often reach out to understand more about the genetics. One of them are people of Ashkenazi Jewish descent. They have very specific risk factors, but then they also don't have other risk factors. So things like that really help us tailor people into the right study and help make sure that we're making the most of that understanding.
Excellent. I have one here. So what would you recommend to families that are hesitant to participate in genetic testing or trials? Is it more helpful to know earlier? And does this participation help find a cure or cause for Parkinson's faster?
This is a great question, and it's one I get all the time. I was speaking to patients just about a month ago, and a gentleman came up after the talk, and he said, "So I've gone through this testing, and I've been tested for this gene. Do I have Parkinson's? I got it last year. Do I need to get more genetic testing?" And I said, "Well, at this point, you've been diagnosed with Parkinson's. Like the genetic test won't tell you that you have Parkinson's. Like that appears to be the case, right?" And he fortunately, was seeing a movement disorders specialist and had a good care team that was helping him. But I did point out, I said, there are certainly studies that you can be involved in, especially because you have a relatively rare mutation. He happened to have some paperwork with him. Often people have some ideas. And so he shared that with me. And I said, okay, so I will tell you that based on the rarity of this, you have gotten pretty sophisticated genetic testing, right? Like this wouldn't come up in some very limited testing. You'd have to have pretty specific testing. So you probably don't need more testing to inform you, but it may tell you a little bit more about your future in the disease. It's something certainly to talk to, again, a genetic counselor about. But also, if you have a family or are thinking about having a family, it may affect how you want to talk to them about the disease, whether they may have a potential risk, whether they want to be tested now or in the future. Because again, in addition to those relatively rare versions that are caused by a single gene, single mutation that happen quite young, there's a lot of them that may come up later, or again, may tell you a little bit more about your need to be out in front of things and get exercise and be careful of your health and things like that.
Wonderful, thank you. That's a really good answer. So I have 3 more questions here. So can you talk a little bit about the Parkin gene and its role in young onset Parkinson's disease?
That's great. I had to really cut it down. There's a lot of these stories that I would love to tell, but I couldn't tell all of them day at a time. So Parkin is another gene that has long been tied to Parkinson's disease. In fact, many of these genes have a different name, right? So for example, synuclein, right, is one of the Parkin genes. I think it's actually Park1 in its code name, and Parkin is another. And so Parkin is a gene that is considered to be something called an E3-like ubiquitin ligase. It's another form of enzyme. Too much for today, but I will just say that but there's a gene called Parkin and there's another gene called PINK1, and those two together are a great example of how, again, two different gears are known to interact together biologically. And so we now understand that mutations in those genes tend to affect the same pathway. So it is tied to young onset Parkinson's disease. That's one thing that we know about Parkin mutations. But it is also relatively rare in terms of the broader causes of Parkinson's disease. So there are a number of active studies that are going on, and I think that there may be a drug trial that is opening up that is a therapeutic based trial that is looking at testing a specific therapeutic within people with Parkin risk factors in their genetic history. So again, a great example of how it is starting to be the case that this may eventually change your treatment trajectory. It may change your understanding of your path through the disease. Young onset Parkinson's disease is an area that I think is really fascinating and one that we think is really important because these are people that are going to live with the disease a long time. So again, great question. Thanks, and hope that was helpful.
Wonderful. I've got two more, hopefully kind of quicker ones here. So the first one is, has there been any definitive link found between like Parkinson's, Parkinsons Plus syndromes, and other autoimmune diseases like psoriasis? Yeah, is there a link there?
Yeah, it's a great question. So I think the definitions of Parkinson's Plus is maybe a little hard to tie into this discussion, but it is an important factor, right? That for many people, Parkinson's is not the only disease that they're going to suffer in their lifetime. And while there are some that are linked, there are others that are not going to be linked. One that I think is really interesting in terms of those links is the link between Parkinson's disease and certain inflammatory diseases, including things like Crohn's disease, right? And so LRRK2 mutations actually appear to have risk variants that affect both your risk for Parkinson's disease and then also for Crohn's disease, which is an intestinal disorder. And we know that inflammation is a key piece of that. There is some evidence that exposure to non-steroidal anti-inflammatory drugs, these are over-the-counter drugs like Tylenol and ibuprofen, things like this, may reduce your risk for Parkinson's in some way. Obviously, we would want people to talk to their physicians about it. It's not necessarily so strong that you would change your routine about that. But that is, again, a clue that this inflammatory pathway is super important. And obviously, inflammation is also a piece of how cells die in our brains. And so we want to understand that more in better detail, and so that we can help understand how these prevention strategies may affect people from ever living up to their genetic expectations, if you will. Like if you do have these risk variants, we want as few of those people as possible to ever suffer from the disease.
Excellent. And the last one here I have is about the DAT scan. So is a DAT scan the only objective way to show dopamine loss in the brain? And should this be more widely available for diagnosis?
That's a great question. So I'll lead by saying that I'm not an expert in neuroimaging techniques, but I think that we have seen that DAT scan has seen increasing uptake in the clinic. It is a pretty clear indicator of its specific function, which is, again, this dopamine transporter, where that is present. And it does appear that the DAT signal is lost in patients with Parkinson's, specifically looking at certain structures in the brain. And so we know that this is a key piece of it. For most patients, it may not change their diagnosis. There are people actually who have evidence of a DAT deficiency, but do not have disease, right? And so that is another important piece, right? You may not have Parkinson's disease, you may not be suffering symptoms of it, but you may have reduced dopamine. And so that might tell you a little bit, this is one of the things we've looked at within our PPMI study, the Parkinson's Progression Markers Initiative study, to try to understand whether those people are developing Parkinson's disease. Is that why they have reduced DAT signal? Is there something else? Often people with a genetic background do have reduced DAT signal at earlier stages. So we're following those patients closely, and that's part of the reason that we're running that as a longitudinal study.